So today I'm going to talk about astronauts, and every kid, statistically speaking, almost 20% to 25% kids of the United States of America wanted to be an astronaut, and that dream kind of got a huge boost with this SpaceX Demo 2 crew mission. And Bob and Doug, when they finally successfully launched to the International Space Station after almost a decade from American soil, more and more kids wanted to become an astronaut. But those missions were kind of like short-term missions, and they spent some months aboard the International Space Station, and that is not as much risky as compared to the long-term space flights, which is slowly day by day becoming a reality with the successful manufacturing and testing of SLS and Starship by SpaceX. And NASA has already announced the Artemis mission, which is going to be launched by 2024. And to speak in terms of astrophysics or aeronautics, the pointy end is up and the flamey end is down. So it looks more and more likely that we are actually going to moon and to Mars, and we are going to stay. So one of the major factors for these missions are these Mars and habitats and moon bases, and more people are coming up with these ideas. But uh, these astronauts are not going to stay inside these bases forever. They are going to come out, explore, and not only that, but the space flight, the journey itself entails a lot of problems. And in the past 1 or 2 months, we witnessed how space tourism is being popularized more and more, and how general public can get access to space tourisms, and they can fly to space. But it's not such a rosy experience after all, because there are a lot of problems. Some of the problems are very traditional, and we have been knowing them for a long time, like bone and muscle density loss, physical strain, fatigue, and stuff. But with the advancements of next generation technology and bioinformatics, we can now also gain insights into other problems like changes in the gut microbiome, and that in turn affects the health of the astronauts, because we know that gut microbiota is connected with a lot of physiological aspects of a person, like the gut and brain axis. Then there's risk of cancer, the reproductive system issues, neurodegenerative diseases, immune response changes, metabolism problems, and many other problems which we are not yet aware of. So, standing at that point, I tried to locate some data sets that are available freely and publicly to everybody for other studies. So, there are numerous projects, and to be honest, very few associated papers. I have made and collected a few of them and thought of some possible projects like this one, like the zebrafish transcriptome under simulated microgravity. This was one study done in association with NASA. Then there was another data available with Arabidopsis thaliana, which was took to space in Blue Origin's New Shepard and Virgin Galactic's spacecrafts. So, to study what happened to these plant samples under the spaceflight conditions, that is, exposed to radiation, microgravity, hypergravity, low pressure, oxidative stresses, and everything. So, that would be something interesting. Then there's hepatic stellate cells under various types of gravity, like 5G, 1G, simulated microgravity. This particular data was generated at Indian Institute of Technology, Madras. So this is the mRNA expression of DLD and MOLT human cancer cell lines under microgravity. And they reported some transcriptomic changes, again, expression level changes, differential gene expressions and stuffs. And also there's one interesting study which was done using the gut microbiota. And there the scientists found very interesting patterns in the changes in the gut microbiome, and they could actually differentiate the difference of this microbiota change pattern from a radiation induced change and a microgravity induced change. So the authors suggest that there can be very significant patterns which we can notice, which the changes which are caused by radiation and the ones that are caused by microgravity. So these are some of the data sets that I have collected. And one project that I have actually started to look at the data and hover around is this one, the zebrafish transcriptome under simulated microgravity. So let's take a look at that. This is the particular paper associated with it. And they actually studied this positive feedback loop mechanism, which is actually a virus-induced immune response. It involves TRIM25 and RIG1 pathway. And this is actually done with the help of interferon type 1. Interferon 1 is very important in this particular feedback loop. And these zebrafish embryos were cultured in standard gravity and rotatory cell vessel, which actually mimicked the microgravity condition. And then they were harvested and their RNA-seq profiling was done and stuffs. Their survival rate was also looked at, and there were two different types of samples. One sample was which had no treatment, and the other one had the poly-IC vector treatment, which is actually a virus mimic, and it attaches itself with the toll-like receptor 3, and it generates an immune response. So these were the two conditions. So in all, we have 4 conditions. So what they actually found was the TRIM25, which is a RIG1 regulated and an interferon stimulated gene, actually had a down regulation. So this is a very important candidate of this positive feedback loop that is triggered to antiviral immune responses. But due to the simulated microgravity conditions, this was reduced. And since this was reduced, the entire feedback loop was kind of hampered, and that made the cells more susceptible to the viral infection. Interestingly, it was also found that upon rescuing the TRIM25 gene and not letting the expression fall, the feedback loop was unharmed and it was saved from being hampered. So the cells maintained their antiviral immune response capability. So the authors discuss there that this TRIM25 is a potential therapeutic target for maintaining the immune homeostasis under microgravity for astronauts in the future. And till date, there's no molecule agonist identified which can actually bind to the TRIM25 protein. And they suggest that maybe adeno-associated virus-mediated CRISPR activation, that is a CRISPR-A system, using a single guide RNA could be a potential solution, which instead of any small molecule drug or small molecule agonist, we can actually use a CRISPR activation system which can bind itself and activate the expression level of TRIM25, and thus, keeping the workflow going. So there's another interesting question that comes up, which is how does microgravity affect and why does these changes take place in the first place? So again, there are few very interesting hypotheses and speculations. Like there are 3 to 5 papers, I guess, in all, which suggest that simulated microgravity substantially affects the epigenetic modifications. So to be precise, the scientists have observed epigenetic changes in myogenic differentiation process, embryonic development, and T lymphocyte regulation. Also, certain epigenetic processes like DNA methyltransferase 3 alpha has been observed to be downregulated in microgravity. And apart from that, DNA hypomethylation, H3 hypoacetylation, and certain mutational changes have been observed. So the authors suggest that maybe this downregulation is a result of epigenetic changes, which they did not explore, and they said that it would be interesting to see in the future. So this is a data set that we have. As you can see, there are this normal gravity samples 3, normal gravity and treated 3, simulated microgravity 3, and untreated and treated 3. So we have 12 samples in all. And so this was a brief analysis of the data set. I did some PC analysis. So the first PC1 versus PC2 has a pretty strange variance, 90% and 3. But we can see that the normal gravity, both treated and untreated, are forming one particular cluster, whereas simulated microgravity treated versus simulated microgravity untreated is not forming a clear cluster. And upon doing a PC1 versus PC3, we see the simulated microgravity ones are clustering together, whereas the non-microgravity, normal gravity are far apart, and PC2 versus PC3 gives a little variance, so they are all not clustering together, they're all apart. There was one pathway enrichment analysis which I did, and we can see there are 4 clusters, and especially in this one, which is cluster C, we can see a strong differential gene expression, that is the normal gravity, all the ones are upregulated and these are downregulated. So I made a list. So we can see some pretty interesting pathways like embryo development, tissue development, brain development, and so on. There's a lot which we can look into. I also did a very quick differential gene expression, and you can see these numbers, and this is a Venn diagram also. And here I have made a little point, like a little chart of the downregulated pathways and the upregulated pathways. So again, we can see some interesting things like protein transport, reproduction, endoplasmic reticulum stress, tRNA processing have been downregulated, amide transport, and under upregulation, we can see gene expression, RNA metabolism, nucleic acid metabolism, embryo development, and so on. So such exploratory analysis kind of shows that, yeah, there can be interesting things to look into as well further. So what I feel and what I want to pass on as a concluding message is there are many pathways which are affected by a single space flight, and space flight means all effects combined. We are only studying microgravity in this one, but there are many other things which can affect, and these studies are really important if we actually want to settle on any other planet or explore them for a long duration, like for 2 years and stuffs, because otherwise we are actually risking the people, those who are going on this particular mission, because we don't really know what's going to happen to them. Also, such studies might help us explore and understand some pathways which we did not know about so far, or to understand the already known pathways better. Like I have one more example, like the MAPK kinase pathway, MAP kinase, which is pretty important for cancer uh, occurrence, is also affected by microgravity. So that's one pathway which is highly studied, which is pretty much established, but now we are studying under a different condition and many different things might unfold. So therefore that would be also something interesting to look into. And finally, I would suggest, or this is kind of my hypothesis, that what can be other ways in which microgravity affects other than epigenetics. Well, there were 2 studies which really made me think like this. So the 2 ways which I think is changes in protein folding patterns, and that can cause some sort of conformational changes and stuffs, and there could be changes in the cell signaling pathways altogether, and that was fostered because of these 2 experiments. One was conducted by Merck in association with NASA. So this is Keytruda, is one monoclonal antibody, that's again a protein, and you can see that its formation and its structure and the way it behaves completely changes under microgravity as compared to normal gravity on Earth. So on Earth, it really doesn't form a uniform pattern, it's chaotic. Whereas under microgravity, they start forming a beautiful shape. So protein folding or shape, conformational shapes might be affected by microgravity of proteins, because this is a monoclonal antibody. Maybe this is one aspect. Uh, this is a kind of hidden experiment of Merck, so I don't really have any associated paper which talks in detail about this. And this is again the analysis of cancer cell lines under microgravity. So when microgravity ceases to exist, the cell lines start rotating, and once they rotate, they freely form 3D organoids, which are known as multicellular spheroids. So without the application of any other factor, these cell lines start to form this 3D organoid or spheroid structure. But what's more interesting is they lose their cell signaling capabilities, and once they lose the cell signaling capabilities, the metastasis actually comes to a halt. Metastasis has been seen to be stopped because of the intervention of microgravity. So again, that kind of supports the idea that there are certain cell signaling changes which comes into picture when microgravity hits in. All right, so that's all I had for today. A really short accumulation of my studies and ideas, and I would love to hear from you guys what you have to say about it.